Now let's look at iron metabolism. The first part of iron metabolism is we have to understand how iron is taken up from dietary sources. So there's two forms of dietary iron. The first is heme. The second is regular iron that can be taken as a supplement. The dietary iron is heme, with bound iron to it, can be taken up directly by a heme receptor. The iron that is taken up as a supplement, separate ferrous iron, this iron originally starts out in a 3+ ferric state. So this is the oxidized form of iron. In order to take up this iron, the first thing we need to do is to reduce the iron into the 2+ state, which is the ferrous state. The enzyme that does this sits on the surface of the intestinal enteric cell, and this enzyme is called D-site B. This stands for duodenal cytochrome B. The D-site B protein performs a redox reaction where the iron is being reduced, so something must be oxidized. What's being oxidized contributing to this is ascorbic acid, vitamin C. So it's widely known in the literature that vitamin C enhances iron uptake by the intestine. This being proximal in the proximal small intestine, it's in the duodenum, so the proximal intestine is responsible for iron uptake. Now once we have the iron in the ferrous 2+ reduced state, now we can take the iron up through another transporter, and this transporter is called DMT1. And DMT stands for dimetyl transporter, also termed divalent metal transporter. So this transporter only works in the divalent form of iron. It is for this reason why it is recommended that people that take iron supplements for iron deficient anemia do not take calcium at the same time. Individuals who take extra calcium supplements, of course, for osteopenia and osteoporosis, sometimes develop iron deficient anemia. And if these two are taken simultaneously, the calcium can compete for the iron uptake and deplete the iron from being taken up inside the enterocyte. Now once we have the iron inside the cell, the iron is never by itself, and so the iron will have two fates. One is that it will bind to a protein called ferritin to form a ferritin iron complex. The iron that comes in from the heme iron transporter is also bound to ferritin protein. Now, once we have the iron inside the enterocyte, there are two paths for it. One is that it will be utilized by the enterocyte, the intestinal enterocyte, for its own activity. Remember, all cells require iron. Think of the oxidative phosphorylation pathway in all of the cytochromes and iron sulfur centers. Or it can be transported out into the blood. Now, to transport this out into the blood, we have a transporter that will transport just the iron across the basolateral surface, and this protein is called ferroportin. Once the ferrous divalent iron is transported across the basal surface, it must be converted back into its oxidized form, ferric state, 3+, and this is done by a redox enzyme called hephaestin. Now, once we have the trivalent iron, the ferric form on the outside, again, iron is never by itself, this is going to bind to transferrin. Transferrin is the soluble protein made by the liver that is an iron transporter. So the transferrin will bind the iron and transport it, escort it through the bloodstream. Now, all cells, of course, need iron for metabolism. So how is the iron delivered to cells? Well, on the cell surface, we have a transferrin receptor. And the transferrin receptor is a transmembrane protein that binds the transferrin iron complex. This then internalizes so that the transferrin iron complex is bound within the lumen of a vesicle as this plasma membrane internalizes into a vesicle. The next step is that the vesicle acidifies. And it acidifies by a protein that pumps protons inside the vesicle, which is going to decrease the pH. When the pH decreases, the iron will now release from the transferrin bound to its receptor. In this term, this becomes called apotransferrin because it is lacking the bound iron. At this point, the vesicles then separate. The transferrin bound to its receptor returns to the cell surface by vesicle fusion, and the iron is transported into the cytoplasm. Now, if we look specifically at the hepatocyte, which is the largest storage organ for iron, what happens here is that we have to convert the iron by reducing it into its divalent form, going from ferric to its ferrous state. And this enzyme is called ferric reductase. Once the iron has now been properly reduced, it is transported into the cytoplasm where it can bind to ferritin and stored in the cytoplasm of the liver. This now, in the hepatocyte, becomes the iron pool. So during any type of iron need in the body, the iron can be subsequently released from the hepatocyte from this iron pool. Now, once this transferrin bound to its receptor gets back to the cell surface, we now have a pH of neutrality, slightly basic, and this causes the transferrin to release from the receptor and return to the blood. When it's in the blood, it is unsaturated, and now it can bind additional iron as needed. Now let's take a look here at ferritin. Ferritin is a necessary protein to store iron. The iron is stored bound to the protein ferritin. So as iron levels change, we are obviously going to need a change in ferritin levels. So let's look at gene expression of ferritin. Once we have our mRNA of ferritin, we have just upstream from the start codon to make proteins, we have a protein that is bound. This sequence, the DNA sequence, RNA sequence that this protein is bound to, is called the IRE, which stands for iron response element. This protein, when bound to IRE, inhibits translation of the ferritin mRNA. Now, if we have sufficient iron around, iron transported to the liver, then the iron can be bound to this inhibitory protein, which causes it to release from the mRNA structure. Once this protein is released from the iron response element, then we can start translation of the ferritin protein. Now, clinically, this is a very useful process because what you are saying is that iron levels are directly correlated with ferritin levels. So as iron levels increase, ferritin levels, which is the storage protein, also increases. Thus, in order to measure an individual's iron level in the blood, we do not typically measure iron itself, but we do measure ferritin. And the ferritin levels in the plasma are reflective of iron levels in the body, specifically in the hepatocyte.